Good day to you. I'm Arvin from the Laboratory of Molecular Cell Biology, Faculty of Medicine, University of Tsukuba. Today I will talk about our laboratory and what are the possible research topics that you can do in our lab. Our lab uses budding yeast Saccharomyces cerevisiae as model organism. It is easy to cultivate, handle, and modify. Also, growing yeast is low cost and fast. This means that you can replicate and conduct your experiments with no trouble. Yeast are eukaryotes, just like humans, and a lot of genes are evolutionarily conserved from yeast to human. It means that we can understand the mechanism of cancer or neurodegenerative diseases by studying what happens in yeast cells. Another thing about yeast is its usage in industrial application like production of ethanol or in food industry. It can be used to address pollution or capture carbon. And because of these advantages, yeast are used in large-scale biopharmaceutical application for recombinant protein and drug production. There are three general research topics in our lab. First is the post-transcriptional regulation of gene expression. Next is the signaling pathway regulating the endoplasmic reticulum stress response. And the third one is the prospore membrane formation by vesicle docking. I will start with the first one. When we talk about post-transcriptional regulation, it is about the fate of mRNA. mRNA carries the genetic code for protein production through the process called translation. In the cytoplasm, mRNA are simultaneously translated and degraded to control homeostasis. The degradation is initiated by the shortening of poly-A tail by the CCR4 NOT complex. In yeast, it was found that the mutation in CCR4 NOT complex have resulted to the slow growth of yeast and aberrant expression of proteins. Our lab is trying to understand the mechanism of mRNA degradation as well as the translational repression, especially that CCR4 mutation are found in different kinds of cancer in mammalian cells. Here are the recent publications of our lab regarding this topic. The next topic is about endoplasmic reticulum or ER. It is the organelle responsible for the folding, modification, and transport of proteins. ER stress results from the accumulation of misfolded proteins. This can be caused by infection or altered metabolism. Recent reports have associated unresolved ER stress to various metabolic and neurodegenerative diseases. Our cells have its own mechanism to address ER stress, and our goal is to better understand what are these interactions and how it works. So here you can see the recent publication regarding ER stress response. For the third topic, it is about the Golgi apparatus. It is an organelle involved in the secretion and intracellular transport. During the meiosis and sporulation of budding yeast, post-Golgi vesicles are transported and fused to the spindle pole body to form a de novo membrane called prospore membrane. Our lab investigates how cargo proteins are trafficked across membranes, as well as the mechanism of how new membranes are formed in the cell. Regarding prospore membrane formation, here is the recent publication of our lab. So what are the possible research topics in our laboratory? First is the yeast-based functional assay. It is possible to screen potential genotoxic substance using yeast. It is a low-cost alternative to animal-based genotoxicity test and can be used to understand the targeted gene variants. Another topic is the sustainable cell factories. It is possible to develop the future trends in energy, manufacturing, and carbon capture using yeast cells. Another possibility is to study about disease mechanism. Study how mutated homologs affect the genome instability, mutation rate, disease mechanism, and discover possible drug targets. And lastly, it is possible to optimize the production of biologics. This involves growth hormones, cytokines, and vaccines. So this is the end of my presentation. I hope I have given you some information about my laboratory, and if you have any question, please don't hesitate to send us an email or visit our laboratory. I hope to see you in the future, and thank you very much for your attention.